Some of the participants today may have already met with us in person. However, there are many new people today, so the idea is to give a general overview of optoacoustic tomography, how it works, its applications, and so forth. And then later, we'll be offering additional webinars that are focused on particular applications. Over time, there has been the availability of many different modalities for in vivo imaging. And generally speaking, we've been moving from the ability to gather purely anatomical information with X-ray and also with ultrasound to being able to acquire functional information, also with ultrasound, CT, MR, and PET and SPECT. And then in the last couple of decades, we've also had access to molecular information through PET, SPECT, and optical imaging. But as these different modalities have emerged, they have not replaced the previously available modalities, rather they've been a complement. And we think this will be the case as well with optoacoustics, where we finally have a single modality that is able to capture anatomical, functional, and molecular information simultaneously. The image that is in the lower right of this slide shows an optoacoustic image of a cross-section of a mouse that is bearing a subcutaneous tumor, and we can see this based on the optical absorption of hemoglobin, which is represented by the grayscale image. On top of this, we can overlay molecular information from an injected probe, in this case ICG. But we'll talk more about this experiment later. Some individuals may not be familiar with photoacoustic imaging. The term photoacoustic, for example, is synonymous with optoacoustic imaging. These terms are used interchangeably. But the phenomenon was first described in a paper in Science that was published by Alexander Graham Bell in 1881. To document his findings, he used a very simple setup where he used a mirror that could reflect incident sunlight through a rapidly spinning slotted disc that enabled him to produce pulsed light. He then aimed this setup at an observer who had a hearing tube in his ear, and inside of this hearing tube was an absorbing substance that could absorb this pulsed light. And what Bell found was that based on the intensity of the incident sunlight, the observer could actually hear a different sound. So he called this the photoacoustic effect. But it was not until about 100 years later until adequate lasers, very sensitive ultrasound detectors, and suitable data acquisition electronics, in addition to sophisticated algorithms, really allowed this physical phenomenon to be translated into a viable imaging modality. On the molecular level, we can also describe how MSOT works. We illuminate our specimen with different wavelengths individually. These 5 lines indicate wavelengths that we would use in the near-infrared region. We can tune our laser in 1 nanometer steps, so we can choose the individual wavelengths based on the absorbers that we want to image. The absorbers within the specimen then absorb this light, and the electrons within this molecule go from the ground state to the excited state. When these electrons return to the ground state, they will either give off light in the form of fluorescence, or they can give off a small amount of heat in the range of 1 millikelvin. This small amount of heat then leads to a thermoelastic expansion surrounding this molecule, which produces a pressure wave, which can produce a sound. We can then detect that with ultrasound transducers, and our approach to imaging is tomographic, which means that we place an array of transducers around the imaging specimen. In this case, we're showing an array that has a 270-degree coverage around the imaging specimen, and inside of this array we could place as many as 256 individual ultrasound transducers. The transducers that we use have a central frequency of 5 megahertz, which allows us to have an in-plane resolution of 150 microns that we can maintain at depth. For each of the individual wavelengths that we use to illuminate the tissue, we get an individual optoacoustic image. So in this case, we used 5 wavelengths to illuminate the sample, so we received 5 output optoacoustic images. We then do spectral unmixing, which means that we look at these images on a pixel-by-pixel basis and determine how the MSOT intensity changes as a function of wavelength. And by doing this, we can identify absorbers with a specific absorption spectrum and overlay onto one of these single wavelength anatomical images the molecular information, in this case, as I said earlier, ICG. So the advantage of MSOT then over other modalities as compared to ultrasound, CT, or epifluorescence, if you look at a cross-section of a mouse leg that was injected with a molecular probe, is that with MSOT you can see structural information such as the bones and vessels. You can also see the molecular probe within the sample. And with the other modalities, you can either see one piece of information or the other, but MSOT allows you to see in vivo both the anatomy and the molecular probe. As compared to PET and optical imaging, because we have 150 micron in-plane resolution, we have a much higher resolution. Because we illuminate with multiple wavelengths, we have molecular specificity, and we can acquire data in real time. Our sampling rate is 10 Hertz, so we can see very fast dynamics. And because we do not need to use ionizing radiation, this modality is both safe and cost efficient. The probes that we can detect depend on the laser which we use, which operates from 680 to 980 nanometers. That means that we can see things that absorb in this region, such as organic dyes, Psi 5.5, IR dyes, ICG, alexiflors, and the like. But we can also see bioinorganic nanoparticles, such as gold nanorods, carbon nanotubes, or quantum dots. Imaging in this window also allows us to see fluorescent proteins. We cannot see all fluorescent proteins, particularly those that absorb in the visible spectrum, but we can see IRFP, which is a recently documented fluorescent protein that has a maximal absorption at 692 nanometers. The information we can therefore acquire with MSOT is anatomical, based on the optical absorption of hemoglobin, functional, because we can differentiate oxygenated hemoglobin from deoxygenated hemoglobin, which allows us to calculate oxygenation. But we can also acquire functional information because we can do region of interest analysis that lets us see perfusion in different regions of the body, organs, or tissues, such as tumors. Because we illuminate with multiple wavelengths, we can therefore then see molecular information, such as targeted probes or fluorescent proteins. And because we capture data at 10 Hertz, we can see kinetic information, so we can document pharmacokinetics and also image in multiple parts of the animal, so we can document biodistribution of injected probes. Our approach to imaging is both preclinical and also clinical. From the preclinical side, we have a proven technology with installations around the world. There has been a huge interest by both pharma and academia, and as we'll talk a little bit about today, we've documented a wide range of applications. But moving forward, we're also looking towards clinical applications. We have prototypes already in development that are handheld and connect directly to the preclinical system. We're also working on miniaturizing the MSOT technology so that we could use this in an intravascular approach, for example, to look at atherosclerotic plaques. And this will really add functional and molecular information tools to the arsenal already available to practitioners in the clinic. This video shows the preclinical MSOT imaging device. The unit itself is about 2 meters tall, 1 meter wide, and 1 meter deep. What you first see in this video is the screen, which will show you in real time both the single wavelength image that's being acquired at the moment, but also a multispectral preview in real time that shows you the distribution of molecular probes. To do the imaging, we then put the animal into an animal holder, which is depicted here. This holder provides anesthesia, gas anesthesia to the animal through this arm, which is delivered to the animal through a nose cone. The animal is then placed into this holder, and then the entire holder is enclosed in a very thin, loosely fitting cassette that is made of a cellophane-like material. The reason for doing this is that when the animal is placed into the system, it's necessary to put the holder into a water bath because there's a 2 centimeter distance from the surface of the animal's skin to the surface of our detectors. And since we do ultrasound imaging, we need to use water for coupling. But it's imperative that the animal neither gets wet nor is it at risk for drowning. So this cassette is both watertight and loosely fitting so it does not interfere with the animal's breathing. The water inside of the tank is also temperature controlled so we can maintain the body temperature at physiological temperatures. This holder is then placed inside of the tank, shown here, and now you can see the ultrasound transducer, in this case a 270-degree array, inside of which are the ultrasound detectors, and this device also provides the illumination. So as you can see in the next part of this video, the animal is then translated through the imaging plane where it is illuminated, shown here with the red bars, and then we detect the outgoing ultrasound signals. We can then do sequential 2-dimensional imaging, and after the experiment, we can render a 3-dimensional volume. On the next slide, we begin with the applications, and we've divided this part of the talk into 5 groups. First, anatomy, then we'll talk a little bit about cardiovascular research, brain topics, cancer research, and then finally we'll end with kinetics. On this first slide, we show typical cross-sections of MSOT showing 2 different regions of the animal. On the left, we show a single wavelength image of a mouse at the level of the kidneys, where you can see the right kidney shown here. You can see the aorta and vena cava in between the 2 kidneys. The left kidney is depicted here. And because the spleen is very well perfused, we see a strong signal from the spleen as well. The spinal cord is also visible at this cross-section. And then lower in the animal at this cross-section, we see negative contrast due to the presence of the intestines. We can also do imaging at the level of the brain through intact skin and skull. And what we see in this case are strong vessels, in this case the superior sagittal sinus, as well as extension to the lateral ventricles. And then at the base of the brain, we also see small cerebral arteries. Outside of the skull, we can also see temporal arteries at the side of the head, and we can also see facial veins at the base of the animal's head. Moving on to our cardiovascular section, we have first done a proof of principle study while imaging the animal's tail to show that our spectral and mixing can differentiate highly oxygenated structures from structures that have a lower oxygenation. In this case, we can do a blind spectral and mixing, which means that we illuminate at multiple wavelengths and then ask our software to tell us what the most dominant absorbers in our image are. In this case, we've documented 2 prominent absorbers, one in blue that shows deoxyhemoglobin, shown by this spectrum here. The print is kind of small, but the axis here is wavelength from 700 to 860 nanometers, and then on the y-axis we have optoacoustic absorption. And then in red is depicted the signals from oxyhemoglobin. And for comparison, we have these spectra that are available from the literature that match perfectly with the empirically measured optoacoustic spectra. We can then overlay the multispectral information onto the single wavelength image and see that the lateral tail veins on either side of the tail have a lower oxygenation compared to the ventral artery at the 6 o'clock position. We've also done some ex vivo studies to show the potential of MSOT in the context of cardiovascular research. In this case, we took mice that were ApoE knockouts that should spontaneously develop atherosclerotic plaques over time, and we injected these mice with a polymer probe that targets P and L selectins that should be expressed on cells, on inflammatory cells. Mice were then sacrificed 2 hours after injection, and the heart and aorta were excised, and MSOT images were acquired and compared to planar fluorescence. What we see on the left is one view of this experiment. where we see that the predominance of the signal is in the aorta from the fluorescence, which is expected in this model, as there should be the presence of atherosclerosis in the aorta. When this is compared to the MSOT imaging, we see prominent multispectral signals from the same part of the aorta, and we see a marked absence of signal from both the distal part of the aorta and also the heart. We can then image this ex vivo specimen in the other plane as well, and see that we can as well see the predominance of signals in the aorta, in the proximal aorta, and comparatively lower signal in the heart. These particular experiments were done ex vivo because at Biomedical, we are in close collaboration with the Helmholtz Center at the Institute of Biological and Medical Imaging, and at this point, most of our permissions to conduct experiments relate to cancer studies, and we are currently waiting to do experiments like this in vivo. But for now, we can present these ex vivo results. We've done an additional study showing the potential of MSOT to detect atherosclerotic plaques. In this case, we imaged excised human carotid arteries that were taken from individuals who had atherosclerotic plaques. In the first panel, we see an optoacoustic image. This is a single wavelength image that can be used to show the anatomy of a cross-section of this carotid artery. In B, we see at two different levels the RGB image of this same artery, and we see that there's a very good correlation between the anatomy that we can see with the naked eye and the anatomy that we can see with MSOT. We then used a probe that was specific for matrix metalloproteinases, that is activatable, and we incubated these ex vivo slices with this molecular probe and then imaged by MSOT. And what we saw was there was a very specific localization of this molecular probe, which we then confirmed with histology the presence of these signals specifically in the region that was documented by MSOT. And at the periphery of this vessel where there was no plaque, we see an absence of this molecular probe. So we know that MSOT can accurately show us the distribution of a molecular probe within a carotid artery, and this was, for us, a proof of principle that moving forward with a miniaturization of our technology with respect to catheters could really make sense in a clinical setting. Moving on to our next section, we've also done some studies looking at the performance of MSOT in the context of mouse brain imaging. In this case, again, we're showing a cross-section at the level of the mouse's brain, where we can see, again, through intact skin and skull, we can see prominent vessels at depth without the need for any invasive procedures. In the next experiment that we did, we injected a molecular probe into one of the ventricles of the brain so that we could produce a complex distribution of a molecular probe and see how MSOT performance would compare to ex vivo fluorescence imaging following cryosectioning. So this molecular probe, which was ICG encapsulated into liposomes, was injected into the dorsal third ventricle. At different slices in the brain, this dorsal third ventricle is connected to the lateral ventricles on each side and also the third ventricle at the base of the brain. So after injection, this molecular probe diffused into all of the ventricles, and then we imaged this animal through intact skin and skull. And what we see is we could localize the 4 individual ventricles in this cross-section, and that there was a very good correlation with the imaging that we saw and the distribution of molecular probe that we saw through intact skin and skull with MSOT as compared to sectioning this animal and looking at the fluorescence. The utility of this experiment is proof that if we had a molecular probe that would have a very specific localization in the brain, for example to the substantia nigra or the hippocampus, that we could differentiate this structure from neighboring structures. Moving forward, we also did an experiment to show that we can document kinetics in real time. we can actually show a couple of screenshots from this experiment where ICG was injected into the tail vein of this animal. ICG does not cross the blood-brain barrier. Rather, the ICG would transiently occupy the vasculature of the brain and head. So these three screenshots show three different time points within the first minutes of this experiment where we see a rapid increase in ICG accumulation in the brain. We can then draw a region of interest at the superior sagittal sinus and quantify the change in signal as a function of time, which is shown in this graph in the lower left. So at the beginning of the experiment, we see a rapid increase in signal, and then over the next 12 minutes, we see a decrease in the signal. There's a couple of important points regarding this graph. First, the clearance of ICG that we can document by MSOT, again through intact skin and skull, shows that ICG is being cleared in a biphasic pattern. This is exactly the pattern that has been documented with many different modalities over the past couple of decades for ICG following its injection into mice. And further, within this first phase of elimination, we see a half-life of approximately 3.5 minutes, which is also consistent with the half-life that has been measured by other means. So we know that the kinetics that we can see in real time are both accurate and can be combined with imaging in the brain through intact skin and skull. The next experiment that we did looked at the possibility to differentiate oxygenated hemoglobin from deoxygenated hemoglobin in an animal through intact skin and skull over time while we performed a carbon dioxide challenge. These 6 images represent a single animal at different points in time divided into components representing either deoxygenated or oxygenated hemoglobin. These graphs then show the quantification of these 2 oxy and deoxyhemoglobin signals in the superior sagittal sinus over this 12-minute experiment. In the first phase of this experiment, the animal received 20% oxygen, followed by, in the next phase, 10% carbon dioxide, after which we gave the animal 100% oxygen, and then finally gave the animal 100% carbon dioxide. And what we can see if we look at these quantified signals is that when the animal received 10% carbon dioxide, we see an increase in deoxygenated hemoglobin, and when the animal received 100% oxygen, this deoxygenated signal decreased, and we see a related increase in oxygenation at the 100% oxygen delivery point. So we know that we can track changes in oxygenation within the brain, and this could be used as proof of principle that functional imaging could be done in the brain of awake mice, or living mice. In the next experiment, we combined our studies of hypoxia and oxygenation with a model of glioblastoma. Shown here are 2 separate animals imaged at different time points following implantation with U87 glioblastoma cells. In the top figure, this animal was imaged 34 days after implantation with U87 cells, and in the bottom image, when the tumor was smaller, the animal was imaged 20 days after implantation. If we look at the top image on the right, this sectioned brain and skull shows that the tumor was quite large, in this case taking up nearly 1/2 of a hemisphere of the brain. When we imaged this animal on 100% oxygen, we could see at the base of this tumor, we could see high signals from deoxyhemoglobin. When we imaged the animal at an earlier time point, when the tumor was much smaller, we found that imaging on 100% oxygen would not actually show us the tumor. Instead, we learned that we needed to image the animal either with 10% carbon dioxide or on 20% oxygen in order that we don't mask these deoxygenated structures. But when doing the imaging in this case, simply by looking at the deoxygenated hemoglobin signals from MSOT, we could see a hyperintensity that correlates with the size, shape, and location of the tumor as shown in an ex vivo fluorescence experiment. So simply by using oxygenation, either at early or late time points, we can document the size and shape of the tumor, and you can track the tumor growth longitudinally. Instead of looking at oxygenation, you could also combine such imaging with fluorescent protein observation. In this case, we transfected U87 glioblastoma cells with the near-infrared absorbing protein I discussed earlier, IRFP, and these cells were implanted into the brain. These animals were then imaged by different modalities to compare the performance of planar approaches from optical approaches to MSOT. So in the second and third panels, we look at planar fluorescent imaging or transillumination that shows us the presence of the tumor, but we don't have any information regarding the depth. So we don't know the size, shape, or location of this tumor. We get more of a yes or no answer as to the presence of the tumor. If we look at fluorescence molecular tomography combined with XCT, which improves the reconstruction algorithm, we can still see the tumor in a cross-sectional image. However, the signal from the tumor is quite diffuse. This is because these signals are generated from photons, and as these photons go from their source in the tumor outside of the animal, they are scattered by the tissue. The advantage of doing MSOT ultrasound imaging is that the scattering from ultrasound waves is 3 orders of magnitude less in tissue as compared to the scattering from photons. We therefore see the highest degree of similarity between the in vivo imaging of MSOT for this IRFP containing U87 glioblastoma compared to the ex vivo sectioning with fluorescence. So MSOT will give you the most accurate depiction of the growth of this tumor in the brain. And with that, the brain section is concluded, and I will turn over the presentation to my colleague, Dr. Wouter Driesen, who will talk to you about cancer and kinetics applications.
Thank you, Neil. Yes, indeed, I will finish up this presentation with telling a little bit about how MSOT can be useful in cancer research and research of kinetics processes. In this first example, we show you how we can use MSOT to assess tumor perfusion. On the left, we see an image of a subcutaneously injected tumor before injection of ICG, 1 minute post-injection of ICG, and 24 hours post-injection of ICG. This MSOT imaging is compared with regular fluorescence imaging shown in the bottom right panel. As we can see in the fluorescence imaging, the ICG signal seems to be very well distributed throughout the entire tumor. However, this does not compare very well with what we observe when we are assessing tumor perfusion with MSOT. We can see that the MSOT signals are mainly located in the periphery of the tumor, and we can explain this by looking at the oxygenated and deoxygenated hemoglobin levels within this tumor. We always get this information when we do in vivo imaging, so we can always compare our results with the oxygenation status within a tumor. And as we can see here, the tumor core is actually very deoxygenated. It's actually turning necrotic, as we can see in this cryoslice validation post-mortem. And this would explain the pattern that we observe with MSOT imaging. And this leads to a more accurate representation of perfusion in a whole tumor. We can also do this in smaller regions, sub-organ level, as we can show here. In this case, we used the orthotopically implanted memory tumor, and we drew smaller regions of interest within the tumor to show heterogeneity. When we draw regions in the periphery over large vessels or more in the tumor core, we get vastly different kinetic profiles. This is basically quantification of MSOT signal over time. This is a period of 10 minutes. As you can see, you can collect data at a very high rate and get real-time information about how well your tumor is perfused at what position within the tumor. Another example is to use ICG for lymph node detection. In this case, this is a non-tumor bearing mouse, but you could do similar experiments in tumor bearing mice, and by injecting liposomal ICG intradermally in the footbed of a mouse, this will go to the first draining lymph node. This is what we show here. We're imaging the leg of an animal, and only one side of the animal gets injected with liposomal ICG, only 5 nanomoles intradermally. And immediately this ICG will go to the first draining lymph node, in this case the popliteal lymph node in the leg. We can clearly see the bone marrow and the blood vessels in red here, which depicts total hemoglobin. And we also see a strong ICG signal in the lymph vessels and the lymph node. So this can be used to detect first draining lymph nodes if you would have a tumor-bearing animal. Another thing we could image is actually processes, depending on the type of probe you have available. In this case, we use the probe that binds to caspase, so this can be used to image apoptosis in cancer. Most cancers are extremely heterogeneous, and with MSOT we can show this heterogeneity. In panel A, I show a single wavelength image of 760 nanometers, and the tumor is circled by a white line here. We can see that there is regions in panel C, this is clear, that are more deoxygenated compared to the rest of the tumor. We can actually see the apoptosis probe accumulating in these regions in the time-lapse video on the right. This video depicts a 1-hour time-lapse video that shows accumulation over time of an apoptosis probe into this more deoxygenated region. In this slide, we show actually the quantification of this accumulation of signal simultaneously with a control dye. So in this case, we injected a caspase probe, caspase binding probe that is depicted in green, and a control dye with a different absorbance spectrum that is depicted in blue. As we can see, the green caspase binding dye increases over the period of 1 hour in the tumor, whereas the control dye circulates for a short while and ultimately clears from the tumor within 1 hour. So at the 1 hour time point, we can then do a three-dimensional probe distribution analysis, as is shown in this three-dimensional rendering. We can clearly see that the green dye, the specific apoptosis dye, is accumulating in the tumor, whereas the control dye is mainly located in the intestines, which is the clearance mechanism for this dye. Third panel shows a validation cryoslice where we can see the green signal within the tumor and the green and blue signal in the intestines because of the clearance of the probe. So this way we can show that we can multiplex, so we can look at a control dye, a targeted dye, as well as oxy and deoxyhemoglobin at the same time. We can acquire kinetic data by acquiring many multispectral optoacoustic tomography time points over time, so you can really get a good idea about the kinetics of the processes you're studying, and you can get three-dimensional volumetric information about the biodistribution of your probe. We then used this same probe to image tumor therapy response. In this case, we imaged the effects of a single doxorubicin dose. We did an imaging before the treatment, and what I show here is a maximum intensity projection of 800 nanometer image, and again, the tumor is circled. We can then image with the apoptosis probe, and we see a small signal in the tumor here. However, after treatment with doxorubicin and we repeat the imaging, we can now see a much more widespread accumulation of this apoptosis probe because the doxorubicin actually induces apoptosis. This can be quantified, and we can see a significant increase in apoptosis signal in these tumors. So the advantage of this probe is that it clears within 12 hours from the body, so this allows for daily imaging of apoptosis within tumors. We've also used MSOTs to track cells. First, I show you a detection limit in vitro. Here we used the agar phantom with intralipid for light scattering, and we placed immune cells, in this case T cells, that were labeled with a dye-IR dye. This dye incorporates into the cell membrane because it's very lipophilic, and it's a very easy method to label any type of cell. We have labeled cells and unlabeled cells in this phantom at 2 centimeters depth, and we can see the MSOT signal. Here we have used 2-fold serial dilutions, and we continue to dilute until we don't see the signal anymore. We can still clearly detect the signal with only 625 cells in the imaging plane, which is a very good lower limit of quantification compared to other imaging modalities such as MRI. Of course, it's more importantly to do this in vivo, and here I show an example of where we took macrophage, another type of immune cells, and we used the same type of dye, but in this case it was a CellView 800 dye, which is commercially available through Lycor. And we labeled those macrophages and injected them systemically and monitored their tumor homing potential over time. In panel A, we see an anatomical cryoslice of this tumor mouse, where the tumor is depicted here, and an MSOT image with, in a jet color scheme, overlaid signal from macrophage. We can clearly see the accumulation of macrophage at 24 hours in this case. And when we look at the spectrum of this signal that we detect here, it's very well correlates with the theoretical spectrum of the dye that we've used to label these cells. We can, of course, quantify the signals over time, and we can see that at the 24-hour time point, we have a significant accumulation of macrophage in these tumors. In kinetics research, it's important to image metabolic organs, and with MSOT, we can do this very well, as is shown in this slide. Due to the very high hemoglobin content in liver, kidney, and spleen, those organs show up very prominently in optoacoustic images. Here is a cross-section at the liver region of the mouse, and we can clearly see the liver with some microvasculature visible. We can see the spinal cord, aorta, and vena cava as well. A little bit lower in the mouse, we can see the kidney. We can distinguish the renal cortex and renal medulla from the renal pelvis, and we can clearly see the spleen, in this case, on the other side. When we then inject probes, either molecular probes or blood pool agents, we can track this over time and visualize their behavior over time. In this case, ICG was injected, and we monitor this over 1.5 minutes time scale, and we can see the ICG coming into the kidneys and circulate. Start the video. You can clearly see the ICG coming into the kidneys and starting to circulate. So you can see this behavior and absorption difference after injection superimposed on a single wavelength image in this case. So using this technology, you can assess kidney function in vivo by injecting a very harmless dye such as ICG. We've also used MSOT to measure probe pharmacokinetics. In this case, we injected a cyanide-based dye intravenously and imaged the jugular vein area in the neck area of the mouse. On the top, you see the visualization of this process right after injection, 5, 10, 20, and 30 minutes post-injection. We then drew an ROI over one of the vessels and actually got quantifiable values that we could correspond to actual circulating levels. With this data, you can then model the data using pharmacokinetic models and get important information about the probe half-life, area exposure through area under the curve, clearance, and mean residence time. So each blue point in this graph represents an MSOT data point, and the green line is modeled. And in this case, we didn't have to bleed the animal at any point, or we didn't have to sacrifice any animals, so you can get a high amount of data points without sacrificing a lot of animals like you would typically have to do to get this type of data. We can not only do this for the blood pool and the blood plasma levels, but we can also look at individual organs. In this case, we had a look at the liver, kidney, and spleen, and observe the behavior of the same probe, how it accumulates in liver, kidney, and spleen. For each organ, you can calculate an in rate and an out rate, which gives you important information about what concentrations that you could expect at what time point. And this way you can optimize your imaging schemes. You can not only do that on a whole organ level, but also on a sub-organ level. In this example from an article published earlier this year, we looked at the clearance of an IR dye, which is known to be cleared through the kidneys, and we observed this over time. What we can see is that the dye first circulates and is clearly visible in the cortex of the kidney, and then slowly moves towards the renal pelvis as it gets filtrated and cleared towards the urine. We can then draw different regions of interest in different regions of the kidney and actually see this differential behavior. In this case, the orange region of interest is drawn in the renal cortex, and we can see an immediate increase right after injection of the dye. And this stabilizes over 30 minutes. However, when we look at the renal pelvis, we can see a lag time because it takes a while for the dye to actually get filtered and cleared. Using this type of kinetics, you can now add a toxic compound that has potential renal toxicity and evaluate if this type of dynamics change. And this way you can evaluate renal toxicity without having to sacrifice animals in real time in vivo. Similar things can be done for gastric emptying. If you would like to investigate if a drug or a compound has an effect on the gastric emptying rate, you could evaluate this by MSOT, as I will show you in this slide. In this case, a bolus of ICG was delivered by oral gavage into the stomach of a mouse, and this is an imaging time point at 5 minutes. The stomach is circled, and you will see the dye slowly moving towards the liver, which is the clearance mechanism of the ICG after it gets absorbed in the body. So at 2-hour time point, there's only a little bit of ICG left in the stomach, and most of it has moved towards the liver. We can quantify these changes over time, so as shown here for the particular mouse that I show up here, but it's very reproducible. We can do this in multiple mice, get averages, and then we can use this to model the data and actually get gastric emptying rates in these mice. We've also validated this by fluorescence imaging. Here we show a fluorescence image at the 15-minute time point and the 75-minute time point. When we quantify those post-mortem fluorescence values, this correlates very well with what we observe by MSOT. Of course, with using much less animals, because for MSOT we don't have to sacrifice the animals to get the data. As Neil alluded to earlier, ultimately we are interested in clinical translation of this MSOT technique, and we are building our first prototypes here in Munich already. Here we show a cup-sized transducer that has 256 elements in a spherical shape. The light gets delivered through the middle through this laser fiber, and we can use this to image vasculature. So in this example, we have a volunteer who is imaging his arm. This box is going to move down. He's tracking the vessel in his arm, and you can see hemoglobin. In this case, you will see his vessels and his microvessels, and whenever he has a mole, you can also see melanin signal, such as here. You have a big bright spot because melanin also absorbs very well in the near-infrared range. On the left, we see the XY direction, and right here we see the XZ direction, which shows depth. As he is going down and follows this vessel, you can see the vessels and the microvessels in the XY direction and the XZ direction. So this clearly has implications for peripheral vascular disease study. You can have a look at the size of the vessels. In this case, we only show a 1 wavelength image. You can also do this multispectrally and actually get an idea about the oxygenation status of the vessel that you're observing. We did a similar experiment in the finger, where the vessels are a little smaller, and the size, the shape, and the direction of the vessels is important in certain disease states, such as arthritis. In this case, we have a healthy volunteer, and you can again see the microvessels in each direction in this person's finger. To conclude, we believe that MSOT is a powerful addition to the scientist's imaging arsenal because it provides anatomical, functional, and molecular information in real time and at a high spatial resolution and even in deep tissue. The range of imaging applications spans wide, and clinical translation is within reach. There's many related publications already available online or by contacting us. We are happy to share those publications with you. And with that, I turn the microphone to Philip, and we will answer any of your questions.
Yes, thank you, Neil and Roger, for all this information. A lot of different applications, a lot of information to digest. So please feel free to shoot questions over. We have a few already. But also, if you have specific application, specific questions, feel free to shoot us an email anytime, and we'll be happy to look at your specific application and see how MSOT can help you. Maybe first question for Neil. You have presented a lot of studies with mice. Is it also possible to image rats in the system?
Thank you, Philip. Yes, it is possible to image rats. We didn't show any of this data today, although this is certainly possible. The illumination and the detection in our system is designed and idealized for tissue that is 2 centimeters in diameter. We designed it this way specifically for mice, but that happens to coincidentally be the same diameter of a rat's head, and also is similar to the diameter of juvenile rats. So we do say that we could do rat neuroimaging, regardless if the rat is young or old, but then we can also image the entire body of small rats.
Okay, going to a little bit more of a technical question. How long does it take to acquire an image, and then how long does it take to scan the whole animal?
I'll answer that one as well. So as we discussed earlier, our imaging rate is 10 hertz. That means we can get 10 separate images in a second. But within that second, each of the individual images take just milliseconds to produce. And importantly, when we go from one wavelength to another, although we didn't discuss this earlier, it also takes milliseconds to tune from one wavelength to another. That means that we can image 10 discrete wavelengths within a second. And if we want to do a multispectral measurement, perhaps with 5 wavelengths, that means our temporal resolution could be half a second. So if you wanted to image a single slice, then it would only take a half a second to acquire your image. If you wanted to scan an entire region, then you need to think about the step size. Our translational stage moves in as small as steps as 100 microns. So if you wanted to image the whole animal in very discrete steps, it really depends on how you want to do the imaging. But at a reasonable step size, it could take as few as 10 minutes depending on how many wavelengths you choose. Alternatively, if you wanted to do a biodistribution study looking at multiple parts of the body, then you could also use our program to set a discrete cross-section for the kidneys, a discrete cross-section of the liver, and a discrete cross-section, let's say, for the bladder. And then you could maintain a very high temporal resolution for these 3 different parts of the body, where it would only take maybe 1 or 2 seconds to acquire each part of the body, and then you could go back and repeat this cycle as long as your experiment requires based on the kinetics of your probe.
Then another technical one, what's the depth of penetration of the laser?
I'll take this one. Basically, our type of imaging is a little different from optical imaging in that we're not as much limited by depth. Depth visualization is mainly dependent on the resolution, on the center frequency of our ultrasound transducer. So this means that we can easily image up to 2 to 3 centimeters depth. So we can get an entire cross-section of a mouse with our current laser. So because we detect ultrasound and not the light coming out of the mouse, we can actually, for mouse imaging, we're not limited by depth.
Okay, maybe another question for Wouter on the specific dyes and probes that you're using. So the question is, the kinetic study was conducted with a special dye. Is it possible to measure pharmaceutical, drug-like molecules, like smaller molecules?
As long as the compound you're interested in absorbs light between 670 to 980 nanometers, we can detect it. Most small molecular weight drugs do actually not absorb in this wavelength, so you would need to conjugate something to your drug that absorbs in this region. But there are examples of drugs that do have an absorbance in this wavelength range, so those could be tracked without using special fluorescent dyes.
A question on the caspase probe. Was the caspase probe an inhibitor probe, and what was the control probe?
The control probe was a dye that wasn't conjugated to any ligand. So it just had similar physicochemical properties compared to the caspase probe. What was the first part of the question?
Was the caspase probe an inhibitor probe?
Not that we are aware of, but I can look into the details of it and let you know.
A couple of questions on the preparation of the animals, maybe for Neil. Could you please explain how the mouse should be prepared for an in vivo experiment? Specifically, should it be shaved, and are black mice possible also?
Yes, I can address this question. So as with optical or ultrasound imaging, If hair is present on the animal, this is going to interfere not only with the lights coming into the tissue, but with the ultrasound waves coming out. So it is necessary to use a mouse that doesn't have hair. So you can either shave the animal or use depilating cream, or you can use nude mice, depending on your application. In the context of C57 black mice, it is certainly possible to shave and image these mice. We've done this many times. If I had in my presence 10 C57 black mice and I shaved them all, what I find just empirically is that one or two of these mice may have very dark spots on their skin where I want to image. These are spots that are very rich in melanin, and this can potentially interfere with the imaging, although this is rather the exception and not the rule. In addition, in terms of animal preparation, I described earlier our animal holder and how it looks. What I didn't mention is that it takes less than 1 minute once the animal is anesthetized to get the animal into the holder, to close the holder, and to get this device into the machine. So this is very easy to use, and we demonstrated this at WMIC last year, and the general response was that this was very user-friendly in terms of animal preparation.
Okay, maybe one more question, going a little bit into the future. Can people do imaging on dogs or rabbits, or if not now, is it possible in the near future?
Yeah, so I'll take that one as well. So this is certainly possible, not in the context of our preclinical scanning, as that's just not the geometry that we set up. However, with the handheld system, it doesn't matter if you're imaging a mouse, a rat, gerbil, cat, dog, pig, or human. This can be possible regardless of the specimen. So it's certainly within the range of possibilities.
Great. Thank you, Neil and Wouter. And we had a few more questions on specific technical issues that we're going to address on a one-to-one basis. So we're going to contact you with answers for those. Thank you all for your time today. And if you have any further questions, please don't hesitate to contact us at info@Biomedical, or write the two presenters or your sales representative directly. And we're looking forward to hearing from you. Thanks very much, and have a good day.